really, I think, a tour de force of signaling in cancer. We've started off with phenotype and epigenetics, and we're going to bring this now all the way back to the clinic and the bench and back again. And so it's with great pleasure that I introduce our next speaker, Bill Sellers. Bill, as I think many of you know, is the global head of oncology at Novartis. Prior to that, he was an associate professor of medicine at Harvard Medical School and the Dana-Farber Cancer Institute. That's where I first met Bill, and also an associate member of the Broad Institute. And so, Bill Sellers.
So I'd like to thank the organizers and Mike in particular for inviting me, and for Tyler for coming to hear me. I am a little worried because I didn't really have anything signaling to tell you about, but I will tell you about translation. And so I thought I would focus my talk on some of the infrastructure systems we've been building that allow us to try to study drugs, shRNAs, and other preclinical perturbations in cancer so that we have a better idea of what will happen in the clinic when we get there. So, the construct is something like this. We have primary human tumors that we want to treat in man, and we have what we call cancer models sitting in a mouse or a plastic dish. And I would say, let's say 7 or 8 years ago, the connection between those two, other than someone called it a breast cancer cell line or prostate cancer cell line, was pretty rudimentary. In fact, I'd say the tool of choice was this high-resolution optical analysis of cancer we called the pathologist, and saying that a prostate cancer was a prostate cancer and a prostate cancer cell line was a prostate cancer cell line. Turns out that was wrong not an infrequent number of times. So we've been interested in correcting at least that connection between the models and the cancers, and tools at the genomic and expression level have become available over time to allow us to do that, either using things like DNA copy number on SNP arrays, Affymetrix expression arrays, and mutation by NGS sequencing. And the Cancer Cell Line Encyclopedia was our first effort with the Broad Institute to create a compendium of cancer cell lines that were annotated in a way that it would allow the data emerging from groups like the TCGA and ICGC to be compared to let us understand how our model systems are not reflective of human cancer. So the first problem when you think about this goal of having an encyclopedia of models is maybe worth just showing or illustrating is the actual scale of the cancer problem. And I think when you're working in one cancer or another, it's easy to sort of get into breast cancer or get into colon cancer, but this is an illustration of, in one way, the scale of the problem. These are the list of cancers if you go to a journal of statistics or something and you look at the epidemiology of cancer in the United States. This is sort of the average list that you would get. So you can already see there's a lot of different cancer types. But I would say what's even more daunting about this is if you take one of those cancer types right here, soft tissues, you can see there's 9,530 diagnosed in the United States in 1 year. And you expand that, that's the number of different types of soft tissue sarcomas that would be listed in the oncology textbook. Now, maybe you think, oh, these are all obscure and no one ever sees them, and who would ever care about these because we don't even make drugs for these. But if you look at 4 of these, five, dermatofibrosarcoma protuberans, gastrointestinal stromal tumor or GIST, liposarcoma, malignant peripheral nerve sheath tumors, and pigmented villonodular synovitis. Each of those is characterized by a pathognomonic genetic feature, and in each of those cases, there's either a marketed drug or a drug in clinical development specifically going after those genetic alterations with a focus on that type of cancer. So now you can imagine the sort of scale of the problem when we seek to model cancer through cell line or xenograft models. So the Cell Line Encyclopedia is a first attempt at that, but I don't want to claim that it's even anywhere near comprehensive because even 1,000 cancer cell lines, which was our goal, isn't going to come close to representing the diversity of cancers that we want to study. So I think as a community and as an oncology group, we need to continue to develop better and better models. So generation 1 of the Cancer Cell Line Encyclopedia was the collection, turned out to be 957 or so cell lines characterized on SNP 6.0 arrays, expression arrays. We now have done RNA-seq and exome analysis. And that information was intended to be correlated with drug sensitivity. So at Novartis, we've been working on studying those cell lines using a robotic system where we profile those cell lines for their sensitivity to several thousand drugs. So this is the robotic system called the Automated Cell Profiling System. It's operated in our developmental and molecular pathology group. These are the cells in the incubator being removed by the robot. This is the cover being taken off the cell culture plate. The plate then goes on to the dispensing deck. This is the pipetter. So this is pipetting in 1576 well format. Then the plate comes off the dispensing deck back into the incubator, sits there for 3 days, it comes out, and then 3 days later, this number of cells are quantitated by cell titer glow. So with that kind of system, you can profile roughly 1,500 compounds in about a 3-month period of time through around 600 cell lines. And you can generate data that look like this, sort of a heat map of compound sensitivity versus, in this case, just cell line designation. And of course, what we're interested in is not the all red, which are completely inert compounds, or the all green, which are basically things that kill every cancer cell line, but really the things that show heterogeneous distribution of effects so that we can then correlate that with either a genetic signature, an expression signature, or other genomic features. So I just wanted to show one example. It actually follows some of Bob's work on crizotinib, and just show you some data for another ALK inhibitor that's in clinical trials known as LDK378. So this is a specific ALK inhibitor. It does not inhibit c-Met as shown here. It has sub-nanomolar IC50s in enzymatic assays and robust nanomolar activity in cellular assays, is very robust in regressing tumors as shown here that bear the EML4 ALK translocation. So the kind of data we're interested in is comparative data like this, where we look at potency of sensitive cells. Here are in red shown the ALK translocated cell lines versus all other cell lines. And you can see the vast majority, there are basically hundreds of cell lines over here that have complete insensitivity to the inhibitor. And if you compare it to crizotinib, here you see some MET translocated cell lines are responding, as Bob mentioned. Crizotinib has both MET and ALK inhibitory activity. So this is the kind of data we can now generate, which gives us a better idea of how selective this might be in humans and where the disease indications are. And in this case, LDK378 went into phase 1 and phase 2 trials, both in crizotinib refractory and crizotinib naive patients. And you can see if you have an EML4 ALK translocation, at least in the early clinical data, the response rate was close to 80%. So I think this is an example of translating the preclinical data now at a scale that's robust enough to sort of set to understand what's going to happen clinically. So with the Broad, we've continued our efforts to characterize the cell lines in other dimensions other than just the genomic dimensions. And in particular, in phase 2 of the cell line encyclopedia, we've done complete miRNA data sets. We've done metabolomics by mass spec, chromatin profiling, which I'll show you some more data on, which histones are extracted and then analyzed by mass spec. With Gordon Mills at MD Anderson, we've done reverse protein arrays on all cell lines, and of course, both at Novartis and in the context of Project Achilles with the Broad Institute, we've been doing functional screens now with shRNA libraries. So first, I just want to show you an example of work that's been done in the chromatin profiling space. And here the question was, does global chromatin profiling, more specifically global histone profiling, reveal new insights into epigenetic modifications? And the reason that was a question is, I think many people thought you might have to study epigenetic alterations with a specific genomic context, rather than just at the bulk histone or bulk chromatin level. So this is really work that was led by Jake Jaffe at the Broad, and I'll get to people at Novartis who were also participating in this. Jake developed a targeted mass spec array in which histone extraction was done, semi-automated biochemical preparation, and then a targeted mass spec approach to look at about 100 different combinations of modifications just on histone H3. So the data so far I'm going to show you is just on H3. We've been doing cell lines, but this is also amenable to primary tissues, and there's standardization, so we get some kind of comparative quantitation across all cell lines. And in phase 2 of the cell line encyclopedia, we've done about 200 to 300 cell lines, but the data I'll show you and that's been published is from the first 115 of those cell lines. So the first question is, does this look like anything, or do you extract histones from cell lines that all looks exactly the same? And everybody's right, you have to study this in the genomic context. And I think you can see from an unbiased hierarchical clustering, even bulk histone H3 peptides are sufficiently different from cell line to segregate cell lines into different clusters. In the work that Jake, Homan Chan, and Greg Kirchhoff did, they identified 6 different clusters. Now the question is that, how did you grow the cells? Was it, the day you grew the cells? It turns out that cluster A was interesting right off the bat because it has decreased histone H3 K27 methylation on mono and dimethylation, but increased trimethylation. This would be a pattern that would be consistent with increased activity of EZH2, and it turned out this cluster was enriched basically for cell lines that had gain-of-function mutations in EZH2. So for example, these are lymphoma cell lines bearing activating mutations in EZH2. Now the cluster on the F had sort of a different phenotype, which is depressed histone 3 K27 trimethylation and elevated unmethylated H3 K27. So this is kind of the opposite phenotype of the cluster I just showed you. And this turns out to be a cluster defined by EZH2 loss-of-function mutations. So certain epigenetic regulators, at least EZH2 in this case, have a strong enough phenotype that you can see the phenotype at the bulk histone level. Obviously, that makes it a lot easier to do things like biomarkers in patient tumors than having to do ChIP-seq, for example. So there was another interesting cluster, which was cluster D, and in cluster D, what you see is a different profile. You see increased mono and dimethylation at H3K36 and a decrease in the unmethylated H3K36. Now it turns out this is a mark that can be enacted by NSD2, and half the cell lines were from multiple myeloma cell lines that bore translocations in the 414 translocation, which is a translocation known to involve NSD2. Now, the first thing that was interesting for me here is that translocation also turns out to be a translocation in FGFR3. So the reciprocal translocation is NSD2, and one translocation is FGFR3. And it's not really been clear which of those two or both of those translocations is the critical driver. And there are, in fact, publications that say it's NSD2. There are publications that say it's FGFR3. This doesn't tell you that it's a driver, but it tells you it has a strong phenotype that's reflective of the enzymatic activity of NSD2. So at the very least, we know this translocation robustly activates NSD2 in a way that's distinct from all the other cell lines that have been analyzed in this panel. But of course, the other question is, what are those other cell lines doing that are associated with this same cluster? Since we have all the exome sequencing data, we can ask, are there mutations in NSD2 or other alterations? And it turned out that all the other cell lines that were in this cluster had a single mutation, E1099K, in NSD2, suggesting that that mutation and the translocation have the same functional phenotype when it comes to the histone profile. So this is an alignment of NSD2 and NSD1 and NSD3. There are 3 NSDs in humans. You can see this is a highly conserved residue. It's not completely conserved in other species. And it turns out this is in the set domain. And if you look at now NSD1 and build a homology model based on NSD1 for NSD2, it turns out that mutation has the potential, and this is a model, so it's not an actual structure, to alter the binding to the substrate peptide. So this is the substrate histone H3 peptide with K36 shown here. And this model suggests that when you substitute the E for K, you might get new interactions with the substrate, suggesting an enhanced activity of the enzyme. Additional experiments were done to actually look at whether the mutant enzyme did or did not confer enhanced activity. And in fact, in enzymatic assays, you can see a more robust activity with the mutant compared to the wild type. And that's demonstrated also shown here with an increased dimethylation of H3K27. Does this, in fact, create dependence in these cell lines? shRNA, DOX-inducible shRNAs were used to ask that question. You can see here in 3 different cell lines bearing the mutation, if you use DOX-inducible knockdown with 2 different shRNAs, you get reduction. I don't want to claim this is complete, but you do get reduction of K36 dimethylation, and in at least one cell line that's associated with a decrease in cell viability and proliferation. We went on to show that that also is true in vivo xenograft models. So that's an example of a story that's emerged from the chromatin profiling from the Cell Line Encyclopedia, and we're of course interested in expanding that effort to the remainder of the set to see if there are other patterns of epigenetics that can be unveiled at the global chromatin state. So the other area we've been investigating, again, both within the Broad and at Novartis, is the use of shRNA screens in the context of the annotated cell line library to try to discover new synthetic lethal relationships in cancer. And our concept here, rather than using isogenic cell lines, we favor an approach using panels of cell lines in which the genetics are known. After you do a set of studies with a library of shRNAs in a given panel, you define a wild-type phenotype by mutation, a mutant phenotype, and then in an in silico analysis, you ask for shRNAs that are enriched in the mutant versus wild-type phenotype. So I'm going to tell you a brief story in which we've used chip-based oligosynthesis to make libraries of shRNAs where oligos are cleaved off an array, ligated into a vector, used to generate a lentiviral pool. That lentiviral pool is then used to infect cells in the Cell Line Encyclopedia. After 5 doublings, those cells are harvested, and the abundance of each shRNA in the pool is counted by next-gen sequencing. Now, we've stayed away so far from genome-scale libraries, trying to focus more on higher depth of shRNA profiling against smaller sets of genes. And in this case, in an experiment that was led by Greg Hoffman in our Pathways group, 17 shRNAs were generated in the library against 385 epigenetic targets, epigenetic genes, or genes that are thought to be epigenetic, and profiled initially against 80 cell lines within the Cell Line Encyclopedia, and then in this case with 17 shRNAs per gene. So very deep shRNA coverage of each gene, hoping that that would overcome some of the signal-to-noise issues that can be problematic in shRNA screening. So as an example of sort of the robustness of this data, we did include known positive controls. In this case, we included KRAS shRNAs, and you can see the degree of depletion of KRAS shRNAs are strongly correlated with the mutation status of the cells being KRAS mutant. So in pink is every cell that's mutant for KRAS. Actually in green, not blue, are all the wild-type cells. And you can see a dramatic enrichment of depletion of KRAS shRNAs in the mutant cells. And if you compare that to a relatively broadly acting shRNA set of shRNAs against a proteasome substrate, you can see most cell lines are sensitive to PSMA3 depletion as compared to KRAS. So this gave us some hope that the selection and selectivity of this method would be robust. So one of the early hits that came out of this is shown here. This is shRNAs against the human Brahma homolog known as HBRM. And here again is sort of the p-value for depletion of shRNAs. And you can see a group of cell lines are highly specific for being depleted of HBRM shRNAs. And we've now colored these for BRG, which is a homolog of HBRM. And you can see the vast majority of sensitive cell lines turn out to be BRG mutant. So HBRM depletion specifically and selectively seems to kill BRG mutant cells. So the first thing I want to show you is, I like this slide because it sort of shows the difficulty of shRNA screens with few shRNAs versus lots of shRNAs. So this is the individual pattern for 12 out of the 17 shRNAs that would have been in that library. And I'm guessing if you go around that, it's hard to pick which one shRNA you'd like to use first, or which 3 shRNAs you'd like to use if you only had to pick 3. And I think the point is that each shRNA has a consistent set of effects on the mutant cell lines, but a different set of noise in all the non-mutant cell lines. And it's my belief that you're overcoming the noise by having random noise in the set versus the on-target activity being conserved across this larger pool of shRNAs. Now, BRG turns out to be mutant in about 10% of lung cancer, so it's relevant to a cancer with significant unmet medical disease. And as many of you know, BRG and BRM are components, and again, homologs of a chromatin remodeling complex known as the switch-SNF complex. So that was very exciting to us because of the notion that if you're impaired or mutant for BRG, you might now be dependent on BRM for the residual function of the switch-SNF complex, and that residual dependence would only be seen in cell lines that were BRG mutant versus wild-type cells. So that you can imagine potentially a broad therapeutic index where wild-type cells have both BRG and BRM. If you could make a selective BRM inhibitor, you'd have potentially quite specific effects in BRG null cancer cells. So we've gone on to look at whether or not you can recapitulate this by sort of more traditional single shRNA genes. Here's two different DOX-inducible shRNAs against BRM, depleting BRM in BRG deficient cells, and that leads to dramatic reduction in colony formation, shown here as compared to BRG positive cells. And if you now look at whether you can induce this synthetic lethal phenotype by first depleting with BRM, then BRG, if we deplete just with BRM, there's no effect. This is in a cell line that has intact BRM and BRG. If we deplete for BRG alone, we have no effect, but now if you deplete both, you can kill this wild-type cell, suggesting that these two are functionally redundant in the context of a wild-type cell. And that you need to deplete at least one of these first to get a synthetic lethal relationship here. And just to finish the story, it appears that the cellular effect of depleting BRM in a BRG mutant cell is the induction of senescence, as shown here by staining with senescence-associated beta-gal or with H3K9 trimethyl mark, shown here under conditions of depletion by shRNA. And this is, this is the in vivo confirmation of that. So those are some examples of the work that we're doing in the Cancer Cell Line Encyclopedia in phase 2, both functionally characterizing those cell lines as well as annotating them for now proteomic modifications, at least with respect to the histone modifications. I'll just finish by talking about a second effort we have to go beyond just using cancer cell lines as models. So I think many people realize cell lines have limitations. They're growing in a plastic dish. There's no stroma. There's no blood vessels. So clearly we would like to not only rely on cell lines for model systems in which to study cancer and try to make predictions about what might happen in humans. So in parallel to the Cell Line Encyclopedia project within Novartis, we've been establishing collections of primary-derived xenograft models over the last 6 to 7 years. And in aggregate now have established 900 models that have limited serial passage. These models are generally around passage 4. They can be frozen as fragments, rethawed, grown up, and used in drug sensitivity studies in mice. We have not fully completed the genomic characterization of the models, but we're in the process of doing exome sequencing, RNA-seq, and copy number alterations. Our group in Shanghai is also working with us on this. So for that reason, we have a considerable number now of Chinese and Asian-derived cancers, including 193 stomach cancers and 88 esophageal cancers and 63 liver cancers of Asian origins. And we're now using both those sets, both in China and the US, to profile compounds. So the first question is, does this, again, have any bearing on human cancer? And we're just starting to get data back on that because, as I mentioned, we're in the midst of all the profiling. Melanoma is one, I think, where you would expect, even from cell lines, to have a fairly good recapitulation of genetic alterations. Here's an example of the common alterations in the MAP kinase pathway, PI3 kinase, cell cycle, switch SNF. Here are the individual genes, and I think in general you can see between TCGA, ICGC data, and the collection of melanoma primaries a fairly good correlation. I don't want to claim that this is perfect, but you can approximate a significant swath of human melanoma. Of course, for other cancers, it's not like this. Breast cancer, as many people know, you have trouble getting ER-positive breast cancers to grow in mice, and we still have a paucity of those tumors. But at least now you know where you have representation and where you don't. So the real question is, you can't grow mice like this in a robotic system. So how can you use 900 tumors in mice as a profiling system? And so over the last year, we've embarked on a set of preclinical trials we call Zeno patient trials or PTX mouse trials, with the notion that we might be able to look at a larger array of tumors growing in mice, but fewer representations of each tumor. And in this case, we call this the 1 by 1 by 1 model, one tumor in one mouse against one dose of a single drug. So we're not going to do 10 tumors per each drug. We're not going to get error bars for each tumor. Instead, we're going to use the diversity of cancers as you would a clinical trial to look at the response rate of a given drug across 50 single tumors in melanoma. In this trial, we look at several different parameters. Of course, progression, which would be no effect, complete response, which is disappearance of the tumor. And then, of course, you can have either stable disease followed by progression, or you can have partial response or even complete response followed by progression. And in these two indications, you have the opportunity to collect resistant tumors and save them. So as an aside, I'll say we now have 200 tumors that are resistant to various drugs that we've put through this clinical trial. So in the first year, we've done 18 different drugs or combinations across melanoma, breast, lung, colorectal, and pancreas. And some of those trials are still in progress, so some of the data I'll show you are not complete. So the first question is, if you're just doing one tumor at a time, how variable is this? And I actually thought it would be really variable. So this is one experiment that begins to answer the degree of variability from tumor to tumor versus drug to drug. In this case, we had two different inhibitors of HDM2 that are shown here. The waterfall plot of all the different tumors, this is pancreas, lung, melanoma, is shown here. And the concordance of each individual tumor responding to two different drugs, these are two different drugs with different structural class, is shown here. And you can see the concordance in response is actually quite high. So even using just single tumors, in this system at least, seems to be fairly robust with respect to reproducibility across two different drugs of the same mechanism. So the second question is, does the result of any drug have any bearing on what happens clinically? And honestly, there aren't that many drugs for which we have both robust preclinical data and robust clinical data. But we included a number in this trial. So the first one is the original standard of care in melanoma, which was dacarbazine, also known as DTIC. Dacarbazine has about a 10 to 13% response rate in patients. In the xenograft trial, the response rate was 13%. In humans, the response rate to DTIC is lower in BRAF mutant patients. And you can see in the Zeno patient trial in yellow are the BRAF tumors. In fact, the response rate to dacarbazine is also lower in BRAF mutant genotype in the xenograft trial. In the clinic, we know that RAF inhibitors, vemurafenib first, and now our inhibitor LGX-818, have a robust response rate in the order of 60 to 70% in BRAF mutant tumors. It's a little hard to see because I'm not showing you only the BRAF, but essentially we get around a 60 to 65% response rate in BRAF mutant tumors in the Zeno patient trials. And then we've recently shown activity of a MEK inhibitor, MEK162, in NRAS mutant tumors. Of course, they also have response rates in BRAF, and we get a distinct pattern of response for MEK162 here. So at least preliminarily in the melanoma trials, I think it actually looks fairly promising that the activity that you see, even measured by response rate, looks quite comparable to what you might see in human trials. We're also, of course, collecting survival data on the mice. I don't want to pretend survival in a mouse is going to be in any way linked to survival in a human, but the odds ratio or effect on survival in terms of magnitude may be quite similar. And you can see here the comparison between LGX-818 at the time where we only had 15 Zeno patients treated versus dacarbazine and the vemurafenib trial that was published by Paul Chapman in the New England Journal. So as I mentioned, we've also been studying various combinations, and some of the combination effects that have emerged now here that are quite remarkable and have also furthered combination trials that are already going in the clinic include combinations of BRAF and MEK inhibitors with a much more dramatic effect now in BRAF mutant tumors, combinations of BRAF and CDK4 inhibitors in which we get again, a much more significant response rate compared to the CDK4 single agent, and then the BRAF plus PI3 kinase inhibitor. All three of these are actually quite positive in terms of increased activity in the Zeno patient trials. So two of these are now, these two trials are now ongoing in patients, and we'll see over the next year or so how well this correlates to the results in human clinical trials. So I'm just going to wrap up shortly, but I wanted to comment, because often I get a lot of questions, why didn't you use this model system? Why are you using this model system? And my comment now to those general questions is no model system is perfect, and no model system is totally imperfect. So you may be a fan of GEMS, you may be a fan of Zeno patients, you may be a fan of cell lines. They all have their relative strengths and weaknesses, which is why we want to use all of those tools to the best purposes we can, taking advantages of what each of those model systems brings as a strength, things like throughput, in vivo setting, for example. I'm not going to talk about it, but I'll just close by saying, so I showed you data from the Cancer Cell Line Encyclopedia and the PTX models. The issue that these have is, of course, even the PTX models are being grown in immunodeficient mice. They're human implanted into mice. There's differences in how the mouse stroma will or won't support a human tumor. We know prostate will not basically grow in a mouse. As I mentioned earlier, ER-positive breast cancer, very hard to grow in a mouse. So there'd be two different approaches to addressing this problem. One is to make mice more like humans, which could take a lot of effort, making a mouse like a human stroma mouse. The other would be to use mouse cancers and understand which of those cancers are genotypically identical or similar to human cancer. And that's the approach we're taking. We're taking genetically unstable mouse strains with DNA repair defects, such as the p53 mutant mice or other mice that have DNA repair that get a diversity of cancers, implanting those into syngeneic mice, and the plan is to, of course, sequence those now and find out which of those tumors are similar or not to human tumors, and we hope to have a syngeneic system of mouse tumors that are genetically characterized and similar to human tumors, and that would be the third arm of this translational system. So I just want to close by thanking patients who participate in the clinical trials we conduct. I had not very much time to thank a lot of people who have worked on all of the projects that I mentioned, and I'd also like to thank our terrific collaborators. In this case, the Broad was a huge collaborator on the Cancer Cell Line Encyclopedia, led by Levi Garraway and his colleagues. Thanks very much.